Up next, we'll be hearing from Aliyah Griffith, who is a graduate of Hampton University and is currently at UNC Chapel Hill in the Castile lab, which investigates coral physiology, ecology, and microbiology. So I'll let Aliyah share her screen and start a presentation.
Can everyone see the screen?
Yep.
Okay, and then I don't know if you can see my cursor. Can you see it moving? Okay, great. So thank you for having me. And of course, my name is Aliyah Griffith. I'm a second year PhD student in the Castile lab at UNC Chapel Hill. And today I'm going to talk to you all about hurricane impacts on coral skeletal growth, specifically in the Florida Keys Reef Tract. So first things first, I wanted to outline basically how and what I will be talking about with you all today. So the initial part one is going to be an introduction. So just some of the quick 101 with coral reefs and hurricanes, and basically then going into part two, what we know about them and how they've been changing. Then part three will be going into, how hurricanes are impacting those ecosystems, and specifically how Hurricane Irma is impacting that Florida Keys Reef Tract. Part four, how the corals are then responding and how they can respond to that type of environmental shift. And then part five, what are the next steps for my research? So first things first, coral reefs. So coral reefs are underwater ecosystems made from colonies of coral polyps that are held together by a hard and strong calcium carbonate base. So with the pictures here, I wanted to really dive into the idea that coral reefs are actually these three-part structures. So where you have that really hard calcium carbonate base, and then as you zoom in, you get more of that functioning coral polyp. Then once you zoom into the coral polyp itself, the coral polyp can move independently and use its tentacles to actually catch food within the water column. But then the organism can also revert to a more autotrophic tactic where it can use photosynthesis due to its zooxanthellae, which again, here's a more zoomed in picture where it's allowed to have that photosynthetic activity within inside the tissue. So coral reefs are essential to this ecosystem, and basically they allow for this sustainability. And coral reefs as a structure for themselves house many different organisms, whether they're invertebrates or fish. They support about 25% of the fish in our oceans today. Moving on to that second image, they also help protect a lot of coastal civilizations from hurricane and storm impacts. So you think of those tropical storms that come along those coastlines, they stop those wave heights and wave attenuation by maximum, usually about 50%. And so it actually helps a lot of those coastline cities and even islands from a lot of that wave impact. The third image here is based solely on tourism. So a lot of that tourism goes back into the economy of those regions and of those areas, where you see people that love to see these beautiful, colorful reefs, and basically they'll spend all their money in those regions and allow them to sustain life practice and any type of civilization. And then that last picture represents medicine. And so there's a lot of research that's going into these coral reef ecosystems because they have properties that allow for medical advances, specifically in cancer. So coral reefs are at risk. And so this is not only a local stressor to them, but also a global stressor. And so the first one, or I guess this top kind of section with 1, 2, 3 are more of those local. So you have your point source pollution issues, your coastal remodeling. So when they're dredging and they want to create, the sandiest of beaches or even beautiful condos, they end up going into ocean civilizations and basically taking out entire reef structures. And then of course, overfishing. And so as you take a lot of those fish colonies out of the water column, it ultimately impacts the coral reef ecosystems. Then going into those three bottom factors, so 4, 5, and 6, you have acute disturbances. So when you think of acute disturbances, you can think of it as a very short period of time, but a high impact event. So your hurricanes and your cyclones. You then have number 5 with ocean acidification. So as CO2 is absorbed into the water column, you have a lower pH. And so ultimately that starts to attack that calcium carbonate structure and starts to disintegrate and eat away at that hard substrate. And then lastly, increase sea surface temperatures. And so today I'm going to focus more on those acute disturbances and increased sea surface temperatures. So when you think of it from a global perspective, a lot of us know about these sea surface temperatures. So we know that there's some aspect of climate change, of global warming, but a lot of people don't know about the specifics and really how it impacts certain ecosystems, and specifically how it also goes into a lot of those hurricane changes. So today I'm going to focus more on the aspects of hurricanes and how they're created and are changing over time. So with this increase in sea surface temperatures, hurricanes are known to form not exactly on the equator, but a few miles off of that coast. And so when you have this warm water that's driven with these dense atmospheric conditions, then as you move away from the equator, the Coriolis effect comes into play. And so it creates these cyclones or these hurricanes. And so they can only move within a certain range of temperature. And once it gets into cooler water, it loses its strength and its ability to sustain itself, and it eventually dissipates. Once these temperatures start to shift and they expand further across the globe, it then changes the range of where and how often hurricanes can form. So of course, they're still not happening exactly on the equator, but as soon as that Coriolis effect kicks in, it can now travel further and longer and even sustain its strength based off of these sea surface temperatures. So one paper that goes into a lot more detail and explains it fairly well is Potent et al. 2020. And so basically they looked at it from a size as well as a strength perspective. So when you look at how big these hurricanes and cyclones are, specifically in regions that are prone to have coral reef ecosystems, there's it's still about maybe the third highest would be in a very frequent category. But as soon as you look at it from a strength perspective, the highest in frequency happens to be the strongest. So basically these are telling you that every 5 years or less than that, there's going to be a very strong storm. And so when they combine the factors of how big the storm is and then how strong the storm is, they saw that these big and strong storms will then occur about every 10 to then 31.5 years. And so for our lab specifically, we usually work between Florida, Panama, and Belize. We wanted to see how it was specifically impacting the US. And so when it came to Florida, they actually broke it down into ecoregions. So they looked at the Bahamas and Florida as one region and showed that it had one of the highest at risk. And so when they looked at big storms, they said that the frequency would be about every 4 years. Then when they looked at the strength of these storms, the very strong storms would occur about every 3 years. And then when they looked at that big plus strong storm, it would happen about every 6 years. And so this puts it in that red zone of having very frequent, big and or strong storms. So based off of this fact, we wanted to see exactly how that would impact that skeletal growth of coral reefs. So when looking at the Florida Keys, we already had coral cores that we were able to collect in 2015. And so once Hurricane Irma occurred in 2017, which was a category 4. So again, you can think back to that big and very strong storm that ended up occurring. We applied for a FAST grant and were able to go back into 2019. So the reason for the size differences in these cores pictured here is that in 2019, we went back and obtained microcores. So some of these cores came from the exact same coral reef heads, and then others were found from corals within the same vicinity. And so the 2 coral types that we decided to work on were Siderastrea siderea, which is down on the bottom left, and then Pseudodiploria strigosa, which is down on the bottom right. And so the common names for that would be your starlet coral and your brain corals. So the overall objective for this project was to investigate those impacts of Hurricane Irma on the skeletal growth, and then looking at their overall growth recovery based off of their locations in the Florida Keys Reef Tract. So the hypotheses for this project were to see how Hurricane Irma would impact the skeletal density. So the idea is that it would increase the skeletal density, but then reduce extension and calcification. And I'll go into how these three parameters relate to each other soon. And so then when you go into the second hypothesis, the idea is that it'll increase skeletal density, but negatively impact the skeletal extension and calcification based off of location. So the idea is that inshore reef systems are known to have more turbid water and have any shifts in conditions, where offshore reef systems are usually used to more clear water and potentially would have a higher differentiation or a quicker reaction to any change. And then for that third hypothesis, the corals may not have any type of reaction because it might be able to transition into a more heterotrophic lifestyle. So again, when I was speaking of those polyps moving on their own compared to having their zooxanthellae work for them and being autotrophs, that can differentiate their feeding ability and potentially change how they're growing and if they're more at risk for food availability. So how we process these corals is that we take them to the biomedical lab on UNC's campus. So here you can see me placing these microcores into the CT scanner. So very similarly to our bones, where if you broke or fractured your bone and you basically go to the hospital and they scan it, you can see different deviations or any changes in those structures. And so corals are very similar to that. So the way we like to usually compare them is like tree rings. So how you can cut down a tree and count the rings and see how old they are, we can do the same with these scans and coral heads. So once we go and take a core out, we can count the rings along the core line to see how old it is and how throughout the years it's differentiated in growth. So here's a picture. It's an older processing picture of one of the microcores that we have. So you can see right where that blue arrow is, there's a pretty low kind of resolution where it's a low density band and a high density band. So you can see how they're kind of coupled along these lines. And so basically that low band and that high band indicate for 1 year of growth. So once we have those coupled units, we can count back the years and see, how far apart these lines are, see if there's any increase or decrease in growth, and then also see the differentiations in the actual coloring or shading. And of course, with analysis or with help from this program, we can analyze to see that density. So when looking at that density and extension, when you multiply them together, we get calcification. So the microcores that I showed earlier, 13 of them were able to come exactly from 13 of the previously cored coral heads. And then of course, due to disease or misplacement, 20 were then taken from newer colonies that were within the same vicinity. So in total, we were able to have 33 cores. So the analysis that I'm showing you here, or the pre-data, as I like to call it, are from 20 out of the 33 cores that we were able to collect. And so this data is basically from more raw data and hasn't been added with any type of ANOVA or t-test yet. But I wanted to show kind of that initial interaction between these sites. So when it comes to the inshore and offshore locations and differentiation in coral skeletal growth, you can see it based off of these three parameters I spoke of before, with density, extension, and location. And so with density, you can see that there isn't a large deviation when it comes to the overall growth or output. But when it comes to extension, based off of its location, there is usually less extension in those inshore reefs compared to offshore reefs. And then when it comes to calcification, you can see that combination of no change, slight change being combined into that calcification rate. And so then the yellow arrows that you see here are just showing you when Hurricane Irma hit. So you can see that there's a slight increase with density after Hurricane Irma. There's a slight decrease after Hurricane Irma with extension, and then again, a slight decrease after Hurricane Irma with calcification. And so again, just so to really ingrain it, as you see these changes, it's because you are combining, that density and that extension to get that overall reaction with calcification. So then we wanted to look at the coral growth on a yearly basis, or yearly difference. And so we saw that some of the more significant interactions was when we pulled out the specific species based off of where it was located. And so specifically the Pseudodiploris strigosa, we saw on offshore, actually was able to increase in density until Hurricane Irma hit, where then it actually decreased. Where most of the other corals, no matter their location, actually slowly increased after Hurricane Irma hit. Then when you see with extension and calcification, there weren't any large deviations over the years in those two growth parameters. And then again, 2017 is just trying to show you when Hurricane Irma hit. So you can see that, again, change or lack thereof post Hurricane Irma. And so in conclusion, when you're looking on that specific location aspect, so those inshore versus offshore interactions, there are significant differences according to that shore location, but very small impacts when it comes to species. When it comes to that pre versus post idealism about when Hurricane Irma hit, there's a pretty solid baseline when it comes to the growth rate. And so it didn't change drastically over 5 years with most of the categories. And then when you start to compare species, there didn't seem to be a huge impact with just this species by itself. But once you included that shore location with species, there was a larger differentiation in impact along those growth patterns. And so going back to the hypotheses I stated earlier, Hurricane Irma will increase skeletal density, but reduce skeletal extension and calcification. So when you think about this, this fact, there was a higher density after the storm, but no difference in extension or calcification in the species and location model. Sadly, though, there was no evidence really showing that there was an increase in density. But when it came to the offshore or the species that were located in the offshore region, there did seem to be an increase when it came to their extension rates. And then the last was a bit inconclusive because there is a pretty solid baseline rate of growth over these years. When addressing all these parameters, you can't make a conclusive idea of how it's exactly feeding or the way that it is and which it prefers. And so those next steps based off of these conclusions is, of course, to complete these statistical significance tests with the rest of these sampled cores, and then looking at not just a combination of that high and low density band to see that annual change, but to split that in half and see if there's any differentiation between a 6-month period. And then, of course, after all of that is done, to potentially look at hurricane impacts across the Caribbean. So because we have a large amount of coral reef cores from Panama, from Florida, from Belize, we wanted to see if we could pair up hurricanes of certain categorical significances, and then see if there's any type of pattern or change in their calcification deposits as well. And so I wanted to acknowledge, of course, UNC Chapel Hill and our medical facility, as well as the Castillo Lab and all of its members for either hearing my practice talks and or helping me with this project. And then, of course, the National Science Foundation for funding for this project, the Davies Lab for helping me collect the microcores themselves, and for Breams for allowing me to present this to you all today.